for example, it's a critical part of the efficacy of drug antibody ratio. That's a critical quality problem. See, our developer and manufacturer and specialists will watch that. It's easy to characterize the control and does as in or consistent match to match. Same thing as for the products as well. The aggregate products also have the free work to achieve drug substance as well. In addition, other including cell-based potency assays as well. Now, my section 2, I will spend a few slides just to give you an overview on WuXi XDC's ability and capacity in terms of expediting COC service to ADC and beyond. As I mentioned earlier, effective CMC starts with a good discovery. People in Shanghai can actually deliver a project in 8 to 10 months. Discovered antibody sequence from PCC nomination. They can do that. They also use healthy technology to screen the candidates in different formats. Process development is not easy. Okay. And as I mentioned, process scientists day in and day out, they can make a process consistent in small scale and large scale. So they can also, it's hard to not use that technology in science. And we can do that. Scientists can do that. They can make a process not step. That's critical for the cost of goods of your product. Especially under the geopolitical times and where the development region, if you have to ship hundreds of liters of very expensive resin to China and make a product and ship that. And all scientists can do that. They can skip that purification with actually just with TF, UFDF, a filtration method versus to purify in a favorable way. To impurities. And we have also scaled up many of this computation chemistry, including the random chemistry, including our IP, WuXi, including the quick chemistry, which actually won the Nobel Prize. And these enzyme acidic chemistry. And therefore, I'm going to skip it. It has its advantage in and it has the advantage of without engineering into actual sequence into molecule. About 15 months, we're very proud of our progress in leading the industry. And the sequence of antibody to ADC IND, but still the top speed in the industry. To do that, we want also to thank our colleagues in the small molecule field. Okay, they are working with the large molecule scientists in terms of development, manufacturing of the linker payload. And they also work with this products in DMF master file. And also, I want to show very quickly that our CMC team, without their support, none of the CMC specialists or lead actually are located in the US. Um, some are located in Europe, and some are located in China. They are the people actually behind the scene, around the clock, actually, uh, uh, coordinating with the scientists at your cell machine to advance your project out. It's first project after 4 to 6 months, as I mentioned. Case study, I'm going to very quickly showing for bispecific ADC. Many of the bispecific ADC, as I mentioned, aggregates. There's a problem with and it's also an antibody aggregates. So we have the same problem. ADC. Our scientists can actually develop processes with ADC within 15 minutes to launch an R&D. As demonstrated the graph on the right side, and we can develop a process can develop purity of 95% of the drug conjugate. The so-called cloud appearance, and that's not easy to set so. Um, a 30 seconds. I'll just quickly show this case study. ACC, antibody curative program. It's important for honestly, candidates. Without this core intermediate, your LRC is not possible. Uh, um, this is an intermediate that has to be developed and manufactured in our GMP. So many of these challenges may not be commissioner or regular ADC. So the first model, uh, the first model or within months, within hours, start this education. Uh, Animated later and making functioning well and so forth. But our scientists are able to do that. We worked with many of the ACC projects in Shanghai and push them into GMP and are actually putting in R&D. All that capability is beyond traditional ADC. To a lot of people, the second generation or next generation ADC, we actually call XDC. The X stands for infinity, possibilities beyond antibody. I'm going to skip that due to the time. One last slide is our Singapore site is built. Construction as we speak. By the end of this year, it's going to be operational. And early 2026, next year, it will be GMP ready. Actually, strategically located with many other big pharma in the forest district in Singapore. Function is expediting CMC from CRDMO service is the best strategy. Okay, and for you, I thank everybody for your attention. Take a picture.
Our third speaker is Dr. Qiang Qin. Dr. Qin is the Executive Director of Analytical Development Gene Therapy at Novartis. Dr. Qin has been working in the biologic therapeutic development field for more than 2 decades. He has extensive leadership experience in analytical development, quality control, and regulatory submission. The title of his presentation is Analytical Life Cycle Management for Gene Therapy Products. Dr. Qin.
Thank you. Thank you, Fia. Thank you. Thank you. Apparently, it probably has edited my slide after I finalize it. I got cell and gel therapy and technical research. So it should be cell and therapy here. It must be my cat working at night. So thank you and the CBA organizer to give me this opportunity to share with you. And it's so great to see many familiar faces and meet new friends here. So I'm going to talk about analytical lifecycle management for gene therapy product. I think Jane and Sherwin's talk pay a good foundation for my talk. Next slide. Do I control here? Yeah. Same disclaimer. All right. So I will first go over a very high-level introduction of cell and gene therapy. For some of you already familiar with the content, please bear with me. Then I will talk about the challenges and opportunity we're facing during early development. I think as well that to begin with, this will showcase that. I will follow that with 2 case studies to see how early challenges, opportunities impact the later life cycle management during the development. So gene therapies roughly are divided into 2 categories. One is ex vivo, which means the cells are taken from the patients. Then the cells are treated with viral-based vectors or non-viral based genetic material to put the gene of interest into the cell. Then cells are reintroduced to the patients. So that's ex vivo. The CAR T therapy is an example of ex vivo therapy. Then the in vivo category, which means the viral vectors and the genetic material are directly injected into patient. So this transduction of the cells actually occur in vivo in human body. So there's no external processing of the cells. So examples of the in vivo therapy are AAV, adeno-associated virus-based gene therapy. There are, I lost counting of the number of approved, probably 8 or 9 already approved on the market. And the recent emerging CAR T is also a hot topic under this category. So just a very quick introduction for adeno-associated virus. So this virus is about 20 meters big. It is a protein-based shell called capsid that's composed of 60 proteins. There are 3 proteins, but it's really from one gene, a different truncation of one gene. Overall, it's relatively low immunogenicity, and there's a pathogenic effect of this virus. And different serotype can confer a different specificity for different tissue, different organs. So if you want to target a specific organ, you can choose a unique serotype. And we have a company that's specializing in screening serotypes to target a particular tissue. Then it can cross barrier, which means it's a great candidate to treat neurodegenerative disease. And the current best-selling gene therapy drug, right, Zolgensma, is under this category. It can deliver up to 4.7 kb DNA in this by this virus, and it's not integrating for the most part. Although there are some data showing that a small amount does integrate into the genome recently. So therefore, it can express a protein or inhibitory RNA or a genetic genome editing tool for other usage. So if you are in this field, you know that we have some setback in the past couple of years in cell and gene therapies, right? Patient deaths, some big company decide to withdraw this field, not because for technical reason, and some of them just because for the size of the market, and also because of the pretty lengthy, complicated regulation process. And this has been a topic in the just a couple of days ago with FDA. It's glad to see the agency is open to hear these opinions. So I do believe this field continue to have great promise. So you can see there are lots of things in the pipeline. All the green ones are in vivo, and the red ones are ex vivo. So this slide just show very quickly the size of AAV and also the complexity of this molecule compared to other modality like small molecule and insulin or typical IgG antibody. You can see, right, aspirin is 180 Dalton, where IgG is at 150 kilodalton. But when it comes to AAV, it's a 5.1 Dalton. So it's a huge sized modality, and it's protein and DNA in it. So from an analytical perspective, you do need quite a bit of assays to probe different quality attributes. Some of the critical quality attributes, some of them are still potential CQAs. There are still lots of unknowns for this modality. So this table shows a typical testing panel for AAV product in GMP lab. It's about 30 to 40, right, depending on the company, versus a typical antibody, probably in the order of 15 to 20. So you can see it's a lot. It's a lot of assays that you need to perform before you can release a lot. So next, I'm going to talk about the common challenges, difficulties, opportunities we face during the early development for this particular modality. Although the principles apply to other modality as well, but there are some unique challenges with the cell and gene therapy. So the first one is that timeline is rushed. And as you just heard from Shiwen, right, some company use CDMO, and Shiwen said day in, day out, they are working on it, right? So if you are in the company, you always keep your competitors in mind, and also patients are waiting as well. So the timeline is normally rushed. Probably 12 months, 15 months, probably people would expect, right, 6 months, 8 months from the initial DNA to IND. Of course, resources are always limited. don't have unlimited resource. This is about cell and gene therapy. Even sample availability is limited, right? As the Chinese saying that 巧妇难为无米之炊. So even the smartest wife cannot cook without rice. So lots of times, analytical actually waiting for process to generate a lot of samples for us to use to develop method. And it's very costly to generate material in the beginning. Third one is because of all this reason, the assay typically is not robust. First, the assay itself is complicated. Lots of times, it's one person or one lab's show. So you can hear that, yeah, we have such a great assay, but a couple of people in the lab can perform it well. When you transfer to different lab, different person may have difficulty. The first bullet point, I think Jane probably, we work on similar things before together, right? The agency always asks us to do more, expect us to do better. And at early stage development, your action routine for the agency to let you go ahead with a clinical trial, and also your company's, I don't know, C funding, whatever it's called, right? IPO, depending on the clearance. So very eager to agree to pretty much whatever the agency wants you to do, which makes perfect sense. But you need to keep the end in that down the road, right? The result of this, there will be some change because of this down the road. I think James' presentation talked about the topic very well, right? Perception that change is important possible in the GMP world. I think James' slide shows it's definitely possible, but very, very complicated, very lengthy. But it's possible. A big lesson we have learned, a couple examples I will show later, is that method development, we incorporate very limited variability. For example, maybe we just have a couple lots of critical reagent, and even the samples are limited, and the metrics are limited as well. And analysts are just limited by the number of the most experienced person in the lab. Because of that, data we generate early on to represent the long-term performance for the methods and the assays. And the specification we set based on this early data may be too low because it's limited by the manufacturing history and by the testing history. And the cost of that going to be huge down the road. The last one is just the overall lack of experience. This is apparent in the CGT field because most of the people in the CGT workforce come from academic background, from Cancer Research Institute, from Gene Therapy Research Institute, so industry experience. So lots of very advanced technology are being explored, used for to develop GMP release assays that is actually not a good practice for QC GMP friendly assay. We actually for to most assay, but simple robust assay. That's the most important. We're not looking for the most sexy technology, not the coolest technology. Okay, as I mentioned, right, so because of all these things early on, we should expect change. Change will occur during the development process. Next, I'm going to talk about two real-world examples, just show, right, how we can change the specification and the method based on data. And based on this experience, right, how you can think about method development on to avoid this, not necessarily completely avoid these issues, but to make this change easier down the road. So the first example is that specification change of impurity because of the acid drift. So as an analytical person, right, the one thing I want to hear is that it's your master issue. It's not process, not manufacturer. The people who are left smiling, I think you know what I mean. But unfortunately, this case is turned out to be, it was the FC. So in this situation, after June 2019, we are making this gene therapy product. But you can see in the figure, right, in a short period of time, we are having thermal loss of the material out of a specification because of a higher impurity level. That's not a good thing in manufacturing. And the VP, the CTO, they are worrying that if we don't know the reason of this out of specification, why we are continuing manufacturing the product, because all the product we are making could still be out of spec, could be down to the drain potentially. So luckily we have some stocked, so we can afford actually to pause the manufacturing process before we can the root cause. So we can imagine the stress, the pressure we had that time, because out of my career, it's the first time that the manufacturing process is held because of analytical issue. So yeah, a team mobilized, a firefighting team started working 24/7. Luckily for this assay, we do have assay control included. Just every time assay, right, this particular sample is together with other samples. So you can always go back to this assay control data on the left, which is a pointer. This one pointer. The one on the left, single assay control, before and after team. Blue dots are before, red dots are after. So you can see that increased on the same sample. The same increase is observed on different samples, but not double times, right, before and after June 2019. So similarly, you can see the trend. After June 2019, that increased. So in June 2019. So this slide just shows if we apply the offset of the assay control to other samples, replotting the applied to population of sample values shown on the left, come looking like population. This is adjustment, still a little bit higher than June 2019, but similar now. So what happened in June 2019? So because of the investigation as unfortunately this case we have a solid evidence showing this is an assay issue, not a manufacturing issue. So to the assay itself, and every day we found out that June 2019 is a time one of the critical regional manufacturers changed their manufacturing site. This case actually brought it back to the US from another country. After that change, we saw this drift. To completely conclude that's the root cause, because by the time we had this investigation, we could not find any change agent, so we could not do a comparison again. But because we have the assay control that show it's the assay shift. So because we have this strong data package, even its impurity level, right, apparently higher, we convince the agency it is the assay, is actually not the level is higher. So therefore, please give us a higher impurity specification. And they did. They looked at our data, they agree with us, it's the assay drift. In addition, right, we did some additional toxicology evaluation, just in case, assume the actual level is a little bit higher. But with this tox evaluation, we convince them, even it's real, it's still not a concern. So yeah, the agency approved, our production resumed. I could sleep better that time. So case study number 2, in this particular case, we were successful in convincing the agency that we can assay from the testing panel. This assay is called titer assay. If you are familiar with it, it's a TCID50 based assay. And, today, but it's closely limited TBS and assay. For example, you could get as high as 1 log difference from time to time. So the question we want to ask is, right, if the assay is so variable, what is the real use of it, right? Is that one time you get number 1, the other time you get 10, what does that tell you? However, this assay has been historically used by academic lab or the industry companies, so it's become an expectation. At the same time, we do assay on the release specification, which is a protein expression assay based on a single image, a single cell image. So this just shows this data. The CV of the 2 assay, right? One CV is a 50% CV, the other one is 1%. Right after this, you can see, well, maybe one assay is higher variable than the other. Again, this just going into the details for the assay, just show a different vector. You get different protein expression for intensity. And measuring that, you get the relative potency. So next slide just show the result from 2 assays at the different storage conditions. The first is -60, which is the storage temperature. So you can see that this assay is pretty flat. The not change observed as expected. Degrees by this image-based protein expression assay, we'll start to see a little bit trend. Then the TCID assay, you still don't see a trend. To 25 degrees C, you see a clear degradation by the image-based protein expression assay, but TCID50 still doesn't tell you much at all. So because all the result of the high availability and indicating capability, we're able to convince the agency this TCID50 based fixed titer assay is really not meaningful, it's not value added. So we got approval to remove this assay from the spec. I can skip this one. So yeah, summary for the case 2. That we all follow the right precedent, what others are doing, what other companies are doing, what the academic lab are doing. But the reality is, we can still use data to justify. We don't have to use certain assay. To conclude my talk, method-specific changes are always challenging, but with, again, the end goal in the beginning, you can prepare for the changes down the road. The regulatory agency, especially like FDA, they're very science-based, data-driven. If you have data to convince them, they will listen. It all comes down to a strong data package. Again, as the case 1 showed, routine samples, assay controls, and consistent critical reagent are very important in troubleshooting a method and to be able to provide a convincing package to the agency when the time comes. With that, finished. Yeah, thank you. Thank you for your attention.
Okay, why don't you pick? Thank you for excellent talks. Please give us speakers a round of applause. So we will start our panel discussions because the time delay, and then we also don't want you guys miss the coffee break. So we'll shorten the panel discussion. One question for each speaker. And then you can ask speakers offline during the coffee break. Yeah, thank you.
All great presentations. Just a question for Jan. Yuan Chen from WuXi Biologics. Thank you for sharing your experience with the global filing and the changes. Just one question about, it's not a main position of yours, but you mentioned the GMP certification. I wonder what's your experience of, say, would the ISO 13485 covers all the 80 plus countries you filed, or it's country by country in terms of?
They usually go by PICs, and depending upon, so if it's a PICS country, they'll take the inspection to go for mutual recognition with acceptance. Some countries specifically would like to inspect, and they're coming. That makes sense.
Good, makes sense. Thank you.
Do you guys have any other questions? Any online questions? Okay.
So those agencies are sales-based. I wonder, like some agencies are, as you said, FDA, but having covered some agencies which is have the scientific justification, but they are still, demanding not accepting scientific justification. Have you globally any cases like that? I think that you're absolutely right. Your experience in this field probably is more than indeed some, especially in the CGT field, right? Because lots of agencies are really as well. Some of them don't have that much background. And I think that's exactly right, the value of this such exchange forums, that the more information we can share, the more opinions we want to exchange with the agency, that will have the impact to change the agency mindset as well. Yeah, I think even as you know, right, the abnormal toxicity, that assay is still being insisted by some countries. That's basically toxicity by injecting your drug into animals. But it's changing, I think, gradually.
Dr. Thank you for for a very comprehensive presentation on ADC drugs. So I have just general question. Since now it's very hot, so what kind of field do you think the ADC drugs can be used for? Like I mean, indication wise.
It's a good question. I remember as a trend. So there's a lot of different problems like infectious disease. There's a lot of indications for the very hard GLP, right? There's people making format as well. But within oncology is still the field. Trend or a harder next generation ADC, as I mentioned, specifically ADC. So it actually the ADC. I think on the ASCO, there's more of the specific also. So very about ADC. So specific ADC is good. As followers, it's going to be in for it's not going to be soon. So yeah.
So we will end our session. I think they said that they have raffle draw in Lakeside. So if you're interested, you can go join the raffle draw. And I think the next session will start right now. Yeah, if you can go grab some coffee and some snacks. Yeah. Thank you. Thank you very much for attending this session.
Hello, 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 hello. Hello. Students suppose say. What do you need? Waiting for the guy.